Survival of the Sickest provides a glimpse into the interconnectedness between disease and genetic and environmental factors. Through a multitude of supported hypotheses and examples, as well as scientific and historic facts, the evolution of our species, as well as many others, is on its way to growing in understanding and may in the future be manipulated to serve beneficial. Hemochromatosis creates an overabsorption of iron by the body, which in turn can create many health conditions such as arthritis and liver failure. Iron acts as a growth agent for even the most basic forms of life and is found within bacteria engulfing cells called macrophages. Hemochromatosis decreases the iron level within macrophages. During the plague of the 14th century, such a condition was considered favorable and allowed for greater survival rates of individuals with the condition, as it kept bacteria from feeding off the iron within macrophages. Bloodletting, normally considered barbaric, is an effective treatment for hemochromatosis as it encourages loss of excess iron. Now we delve into the relationship between diabetes and the Younger Dryas, an ice age occurring some thousands years ago. Grapes, the wood frog, and many other types of organisms have displayed incredible adaptations under freezing temperatures. A similar tactic is explicit in all of them, however. The internal water reserves are either depleted or restationed, and blood glucose levels rise tremendously. Decreased water reserves means a less chance of ice crystals forming and puncturing cells and capillaries, as increased blood sugar decreases the freezing temperature of the blood, and, when supported by brown fat, allows for immediate heat release. Diabetes has also been found to be more common in people of Northern European descent than in people of purely African, Asian, and Hispanic descent, which supports the link between diabetes and the Ice Age. Moving on to a warmer day and age, sunlight has been getting a bad rap recently, but it isn't all bad. It helps create vitamin D, a crucial nutrient for bone health. However, it also decreases folic acid. Folate is extremely important to the cell growth system, assisting in the replication of DNA. Evolutionarily, darker skin evolved near equatorial locations to decrease the amount of folic acid lost as a result of extended direct contact with sunlight. In contrast, light skin evolved in far northern locations to maximize absorption of vitamin D. Vitamin D can be created in our bodies through cholesterol. The APOE4 gene is an example of an evolutionary adaptation that ensures a high concentration of cholesterol in the bloodstream. However, while a buildup of cholesterol helps encourage the creation of vitamin D, it also is cause for greater risk of heart disease and other complications involved in the buildup of arterial cholesterol. Greek philosophers often warned against the consumption of fava beans. This, in fact, may serve as useful to many, especially for those living in Africa or the Mediterranean, where fava beans are heavily cultivated and consumed. However, it is also where the enzyme deficiency favism is most prevalent. Favism results as a depletion of the crucial protein G6PD, a sex-linked trait, which normally acts to rid the body of free radicals, which are molecules or atoms with unpaired electrons that cause early cell death. Fava beans have viscine and conviscine, which produce free radicals. Fava beans in combination with favism create severe hemolytic anemia. These plants, along with many others, have acquired characteristics to deplete a predator's body and protect itself. Plants, just as well as animals, are able to adapt and survive. Parasitic organisms and even infectious viruses have evolved to manipulate both the bodies and minds of bigger organisms. For instance, the guinea worm, which lives most of its life in the small intestines of humans, once ready to reproduce, secretes an acid to create an exit way through the host's skin, which encourages the host to instinctively seek a body of water to cool the burning sensation. Once in contact with water, however, the guinea worm releases its larvae. Another tactic used by parasites is the utilization of intermediate organisms, as does a lancet liver fluke, which lives primarily in the livers of sheep and cattle. In order to move from the liver of one animal to another, its eggs are released through the host's dung, which is then transferred to a land snail, whose slime ants will consume. This is when it gets weird. The larvae will secrete a protein to make the ants suicidal, standing at the tip of a grass blade, hoping to be eaten by a sheep or cattle so that the cycle may continue. The integration of viruses and bacteria into the human genome is a relatively new development in the scientific community. It has been found that they make up as much as 1/3 of human DNA. Up until recently, it was also believed that non-coding segments of DNA, which make up 97% of human DNA, were essentially junk. These segments contain transposable genes, or jumping genes. Jumping genes respond to internal and environmental stresses. When under certain stresses, it becomes essential to survival to adapt very quickly. An example of this adaptation is the observance of flu outbreaks following sunspot peaks. Antigenic drift, or when a mutation occurs in viral DNA, is believed to be caused by the peak of sun radiation as it encourages genetic mutations. We now know that non-coding DNA is important to the past, present, and future of human characteristics. A recent development in the scientific community involves the altered expression of genes without any actual changes made to DNA. This is called epigenetics. Certain compounds can attach themselves to specific genes and suppress their expression. This occurs when environmental factors signal the attachment or detachment of said compounds. An example of this lies in the 2003 experiment when two fat yellow mice that contained the gene agouti were given vitamin supplements during pregnancy, resulting in skinny brown mice. Supplements chemicals suppress the agouti gene in the developing mice. This suppression is called DNA methylation. This results when a methyl group binds to a gene and changes its expression. Methyl markers, established through external environmental conditions, contribute to the process of evolution for when they are passed generation to generation, patterns are established, and characteristics are constructed. Programmed cell death may be responsible for aging. Telomeres at the end of chromosomes act to protect against loss of genetic material, since as a cell reproduces, it loses small amounts of DNA. Because of this, a cell can only divide a specified number of times. This limit is called the Hayflick limit, and it just may be an intended adaptation. We evolved to have a cell reproduction limit to protect against cancer. Some cancer cells employ the use of the telomerase enzyme, which lengthens the telomeres at the end of chromosomes, creating practically immortal cells. New research has uncovered the idea of pre-programmed aging. For example, progeria, an accelerated aging disease, is caused by a mutation of a gene responsible for the laminin A protein that would normally provide structural support for cell membranes, resulting in rapid cell deterioration. Further research may help uncover the secrets of aging and death, as well as help defeat the fundamental tactics used by cancer cells. Conclusion: survival of the sickest provides a definitive connection between what are now considered medical conditions and one's prolonged longevity through a copious amounts of case studies and examples. Evolution is capable of adapting, sometimes very quickly, to environmental threats, creating stronger and more capable populations. It is a matter of the pros outweighing the cons on an evolutionary scale.